Hello, my name is Chloe Woodman, and my project is titled Visualizing Membrane Remodeling Upon Polio Infection. I worked at the University of Maryland in the Department of Veterinary Medicine, and I studied under the direction of Dr. George Belov and Dr. Massimo Ghimi, both of whom work in the Belov Virology Lab and study all things that have to do with polio. So let's get started. Polio has been eradicated for years. What now? Well, actually, polio happens to be a member of the picornavirus family. Many diseases that are common today are also picornaviruses, such as hepatitis A, foot and mouth disease, and the common cold. And since we have vaccines and treatments for polio, it's an excellent tool for learning more about viruses similar to it, and we can use it as a proxy in a lot of research. So specifically, what can we learn from polio? Polio is a virus, which means that it relies on the sustenance of other organisms to replicate. Different classes of viruses achieve this in different ways. And for viruses similar to polio, they form a center for replication within the host. This is made out of key macromolecules like lipids and proteins. We hope to understand how the virus recruits these materials, where they gather them from, and what the replication complex looks like. Understanding replication is really the key to developing vaccines and cures for viral diseases, even those as elusive as the coronavirus. So how do we study polio? Well, lipid droplets and endoplasmic reticulums are potential sources for the matter that make up polio's viral replication complexes. To see how these organelles move, we can track them with something called fluorescent markings. This can be done in a permanent manner with something called a stable cell line that's achieved by a retrovirus, or in a temporary way with something called transient transfection. For this experiment, a stable cell line was developed, but transient transfection was ultimately used to examine the organelles. So the reason we chose lipid droplets and rough endoplasmic reticulums was because, as we mentioned, we know that polio needs lipids and we know that it needs proteins to build the replication complex. And we were curious to see whether these were the sources of those macromolecules. And this is likely because lipid droplets, well, it's in the name, they store lipids, and endoplasmic reticulums are part of the protein synthesis pathway, which means that they are part of the steps for creating protein. Okay, and you can see in the diagram that I have in the bottom right that the rough ER is located adjacent to the nucleus and that lipid droplets are distributed in little bulbs throughout the cell. And this is a simplified model of the cell, just highlighting the organelles that are relevant to my project. So transient transfection, what does that do? Well, it's a method of sticking fluorescent pigments to areas of a cell that you wish to view in detail. For this experiment, we added green fluorescent protein to the lipid droplets of a human cell and red fluorescent proteins to the endoplasmic reticulums of a human cell. In my diagram that you can see on the bottom right, we have a plasmid, which is basically a circular chain of DNA. This plasmid temporarily enters the host cell, introduces its genetic material, and instructs the cell to do things like fluoresce. Now, what happens during infection? What did we find out about polio? Well, these images that we have are images of poliovirus infected cell on the right and a non-poliovirus infected cell on the left. And these have both been marked by the two fluorescent treatments that we have, so there's an overlay of those two treatments in these photos. In the infected cells on the right, it is clear that the organelles have moved to the same location, adjacent to the nucleus. This shows that polio disturbs the position of protein and lipid-related organelles, such as the lipid droplets and the endoplasmic reticulum, to use in its own RNA synthesis at the viral replication complex. And what can we do with this information that we obtained today? Well, knowing so much about what polio uses to replicate will be useful for the scientists that work on vaccines for other coronaviruses. They know that their treatment must prevent the relocation of lipids and proteins. In the process of my research, I also developed a tool called a stable cell line using a retrovirus. That will mark fluorescent signals in human cells in all its replications. So essentially, this can be used indefinitely by the researchers in my lab to track where the endoplasmic reticulum is located in any sort of infection, so using whatever virus you could imagine. As you can see, in a time like this, the researchers that are trying to overcome problems that may seem obscure are actually absolutely essential to the health and safety of the world. The scientists that were preparing corona-type virus research before the COVID-19 outbreak are the ones that will be able to help us regain a new normal life in the hopefully soon future. While I hope to never live through a time like this again, it really only highlights the important work of scientists and virologists who are curious enough to explore viruses you may never have thought of. And with that, I'd like to thank you for listening to me today and thank my mentors, Dr. Belov and Dr. Masamogini, as well as my RP teacher and my internship coordinator, Dr. Tu. Thanks, everybody. And if you have any questions, I would really encourage that you visit the University of Maryland Vet Medicine website, where we have lots of information about the research that's going on at my lab and under the supervision of my colleagues.